Welcome back to Mitochondrial Medicine 2021. Our final clinical trials presentation will feature discussions from Astellas Pharma, Reneo Pharmaceuticals, Stealth BioTherapeutics, and Zogenix.
Hello again, everyone, and welcome to our third and final clinical trial update panel of the day. In this session, I will be joined by representatives from Stealth BioTherapeutics, Modus Zogenix, Astellas Pharmaceuticals, and Reneo Pharmaceuticals. As with the previous panels, each of these representatives will provide a brief 5-minute update from their respective organization on their clinical trial activities and plans. And at the conclusion of the updates, we're going to be joined by Dr. Richard Haas of UC San Diego to discuss some of the challenges associated with recruiting, enrolling, even patient navigation and finding the right clinical trial. I'd like to just remind our audience that all of these panels are available on our virtual poster hall. If you're a meeting registrant, you have free access to that. And all the presentations will be available on demand 24/7 through the end of July for your reviewing. Many of these organizations also recorded a longer, more extended version of their company presentation. So I encourage you to visit the virtual poster hall to see those as well. With that, why don't we get started? Our first speaker is Jim Carr, Chief Clinical Development Officer at Stealth BioTherapeutics. And Jim, I'm going to turn it over to you for your presentation.
So thank you again for the introduction, Phil. I'm going to talk about a clinical trial that we're hoping to start enrolling by the end of this year. Before I talk about the trial, I will talk about a finding that really led to the design of this trial. As I talked about last year, there was a pre-specified nuclear DNA subgroup from our SPRIM 300 trial, which suggested a response in the 6-minute walk test. Particularly noteworthy in the left part of the slide is that the patients experienced the benefit that received elimipritide. However, if patients were assigned a placebo, there was really no observable evidence of a response. So that, in our mind, helps to validate the finding. Again, there was no suggestion of a benefit in patients that had a mitochondrial DNA mutation. It was only the subjects that had a nuclear DNA mutation that exhibited response. Another reason we feel like this signal is valid is because there was an exposure-response relationship that was observed in the nuclear DNA patients, and there was not such a relationship that was observed in patients that had a mitochondrial DNA mutation. On the right part of the slide, and in fact, the post hoc analysis that disclaimer refers to this analysis where we dug a bit deeper into this finding, the nuclear DNA mutation subgroup, and the deeper dive revealed that patients, in particular with a replosome-related nuclear DNA mutation, exhibited an even stronger response. Furthermore, if you then looked at patients that had PEO, or progressive external ophthalmoplegia, in addition to a replosome-related mutation, that the response was even more exaggerated. So going forward, the trial will be enrolling subjects, and I'll scroll to the next slide. Our proposed phase 3 design, and this is proposed because we're still seeking concurrence with FDA on the characteristics of this trial, but the proposed trial will enroll subjects with a nuclear DNA mutation. Again, we're paying close attention to subjects that have a replosome nuclear DNA mutation. And then in particular, we're looking for subjects with PEO. So additional patients can get into the trial, even though they don't have a replosome-related mutation. But again, the particular focus is once again on the patients with the nuclear DNA mutation of the replosome gene. This will be a 1-year trial. It's a 1-to-1 randomization. Patients get randomized to either drug or placebo. It's 48 weeks of double-blind treatment period. People that are familiar with how we dosed the last trial will recognize that we're dosing higher, 60 milligrams instead of 40 milligrams with our first trial, and that's in recognition of the fact that there was an exposure-response relationship observed, so we're trying to optimize the response a bit further. The primary endpoint will be a 6-minute walk test. We're also looking at the 5-time sit to stand, the TUG, and some PROs as secondary endpoints. As I mentioned, it's a 1-year duration trial. with a 4-week follow-up period. And again, after we get concurrence with FDA, the goal is to be initiating this trial before the end of the year.
Okay, thank you, Jim, for that update. And it's really exciting to hear that there may be a clinical path forward for elamiprotide in a subpopulation of mitochondrial disease patients. We'll look forward to more details on that as they emerge. I also want to thank you. I know Stealth is committed to working with investigators involved with previous trials to publish the results of those trials and share that with the community. And we look forward to seeing those publications when they come along.
Yes, we're absolutely committed to publishing the results from SPIN 301, Phil. As we're also pursuing additional publications. As some people might recall, there was a registry that preceded the 301 trial, so we will publish the results of the registry. And we're doing a deeper dive into the subgroup finding, and that too will be another primary publication. So again, this is something we feel strongly about, and we are absolutely committed to seeing these data published.
Great. Thank you again, Jim. Appreciate that. So we're going to turn to our second speaker, Joanne Quan. She is the Chief Medical Officer at Modus Therapeutics, a subsidiary of Zogenix. And Joanne's going to provide an update on their clinical activities. Joanne.
Thanks, Phil, for the opportunity to share some of our exciting work with you, the scientific community, and patients and caregivers. We're developing the investigational therapy MT1621 for thymidine kinase 2 deficiency. MT1621 is designed specifically to restore mitochondrial function in TK2 deficiency. On the left, you see a diagram of a mitochondrion from a TK2 deficient patient. The decreased function in the TK2 enzyme causes the nucleotide pool to be depleted. And since nucleotides are the building blocks for mitochondrial DNA, this leads to decreased amounts of mitochondrial DNA and eventually muscle weakness. On the right is a mitochondrion from a TK2 deficient patient who's receiving MT1621, a fixed dose combination of DC and DT. DC and DT are normal components of DNA, but because of the genetic mutation in TK2, the building blocks cannot efficiently be used to make mitochondrial DNA. Working through the residual TK2 activity, as well as a couple of other enzymes that are not depicted, MT1621 increases and tries to normalize the nucleotide pool, thereby increasing mitochondrial DNA and improving cell function. And this increases survival in animal models. On this slide, you see on the left what happens in untreated TK2 deficiency. For many patients, especially those with very young onset, muscle weakness is rapidly progressive and leads to death from weakness of respiratory muscles. And even in patients with older onset, they may have debilitating weakness and require ventilator support. On the right side, you see some of the results that we've shared at UMDF and other meetings. This is from the retrospective study that we call MT1621-101. In this study, there were 38 patients. All have survived and are continuing on treatment. The vast majority showed stabilization and or improvement, including those with rapidly as well as slowly progressing disease. And indeed, some patients, especially those with younger onset, had really profound responses. And you see a few examples here. 3 patients who regained the ability to walk, and 1 patient who was on a ventilator full-time who was able to come off completely after treatment. We have similar findings in an ongoing study, and any further studies will be based on discussions with health authorities. In parallel to progressing registration efforts in the US and Europe, we have several initiatives that are patient-focused. We have a no-cost genetic testing program with Prevention Genetics for patients suspected of having TK2. And I think very exciting for this audience, we're partnering with UMDF on a diagnostic program specifically designed for mitochondrial disease patients. And I think there's a bit of a teaser for you, Phil, for a subsequent session where you're gonna share that with a broader audience. We're also working to better understand the impact of disease on patients. Patients, caregivers, and physicians are invited to register at tk2d.com to receive more information. The University of Washington is doing a study to learn about the impact of disease as well as treatment on patients. And Andrea Wilkinson from our patient advocacy group is gathering patient stories so we can increase awareness and ultimately improve diagnosis and treatment for this debilitating and often fatal disease. So in summary, 1621, MT1621 is specifically designed to treat TK2 deficiency. It's made of components of DNA and restores mitochondrial function. And finally, we have compelling clinical data showing benefit in patients with rapidly as well as slowly progressing disease. We look forward to bringing this therapy forward to the mitochondrial community. And thank you for the opportunity to share our work with you today, Phil.
Thank you, Joanne, for the update. And indeed, we're excited to be able to share some of the details around the genetic testing program in the next panel discussion after a short break when this one ends. So stay tuned, everybody, for that. With that, I think we're going to turn to our third panelist of this session. That's Gerard Merrick. Gerard is the Executive Medical Director at Astellas Pharmaceuticals, and he's going to provide an update on what Astellas is up to with clinical trial activity. Gerard.
Yeah, thank you for this opportunity for us to present what we're doing on ASP 0367 at the current time. We have a study that started up called Mountainside. And we have our usual disclosures. And what we're doing at the present time is we're starting up a phase 2-3 study, randomized double-blind placebo-controlled study, which will have almost 140 participants. And the purpose is to assess the effects of 0367 on functional impairment and fatigue compared to placebo, and also to assess safety and tolerability. It's going to involve about 15 sites around the United States, and to find a site that'll be starting up near you when they are actually activated, you can search clinicaltrials.gov at NCT 0464 1962. And this just shows a schematic of part of the study. We have a phase 2 part and a phase 3 part of this study. The phase 2 part, the participants will be going on either high dose or low dose or placebo. And then we'll be looking at PK and pharmacodynamic effects essentially in terms of energetics that we see in terms of turning on PPAR delta related genes, because our compound is actually working on a protein in the mitochondria called PPAR delta that turns on genes that improve muscle energetics essentially. And then we'll pick one of those doses on the basis of the first 30 subjects, and then everyone will continue on for a plus 50-week schedule on the selected dose or placebo on the selected dose. And then after that, everyone can go on open label drug as well. So this is sort of the dose selection part in the first 30 subjects. And then for the additional subjects, they'll have a 4-week screening period. They'll be going on the selected dose from this phase 2 portion or part 2 of the study. And then going on either that dose or placebo. And then after the end of that 52-week period, we'll have an opportunity to go on open label drug for an additional 24 weeks before a washout period. So, the usual things will be happening at the study visits in terms of looking at your general health, how your body uses the drug, PK test, how you move your body. We'll be looking at a 6-minute walk test as the primary endpoint, also a video test at home, and then also we'll look at relations between functional improvement and imaging at one of the centers that we'll be using in the study as well. And this is open to adults ages 18 to 80, having a diagnosis of PMM and able to adhere to the study requirements, and then number exclusion criteria as well. And you can see all the additional exclusion criteria on the clinicaltrials.gov site as well. And then, as I mentioned, for more information, you can go to clinicaltrials.gov, NCT 0464 1962 for more information, or you can contact us at astellas.registration@astellas.com or 800-888-7704 for more information. So thank you very much for listening to our presentation.
All right, thank you, Gerard, and thank you for mentioning again clinicaltrials.gov. In a previous panel session, I called this out. This is a really good resource for our patient families to find out more about the clinical trials that are being discussed here today. So please take advantage of that resource. With that, we're going to turn to our final speaker of this session, and that's Dr. Alex Dornbaum from Reneo Pharmaceuticals. He's the Chief Medical Officer, and Alex is going to provide us an update on Reneo.
At Reneo, we're also developing a PPAR delta agonist. And as you know, PPAR delta is a receptor that sits in the cell. And when you activate PPAR delta, you activate genes that are involved in the function of the mitochondria. You also activate genes that may help make more mitochondria. And the end result of that is that you have better mitochondrial function with improved oxidative phosphorylation, and that may help the muscle work better. The PPAR delta receptor, by the way, is present in those tissues where there is more high energy demand, like the brain, the heart, the muscle. So these are the tissues that generally are affected in patients with PMM, and that we hope we can improve in their function. Now, Reneo, the drug we are developing, is a once-a-day oral formulation, very easy to take. And to date, we have had quite a bit of experience with this drug. We've initiated 6 clinical trials with this drug, of which 4 are completed. This is a summary of the trials that we've completed to date. We've done a single ascending dose in healthy people just to look at the safety and how the drug behaves in healthy volunteers. We did multiple doses in healthy volunteers as well. And we did 2 other very interesting studies. We did a study in healthy volunteers where we immobilized the leg to cause muscle wasting and to mimic, weakness in the muscle as you would potentially see in a patient with PMM. And we also did a study in PMM patients. Let me share very briefly about those studies with you. So in the leg immobilization study, healthy volunteers were divided by chance. They were randomized to receive either Reneo or a placebo, and they took it for 4 weeks. During the first 2 weeks, they had one of their legs, it was the left leg, immobilized. And that causes muscle wasting and decrease in strength in the muscle. At the end of 2 weeks, we removed the cast and we measured the muscle strength in that leg. And in the right-hand side, you can see what happened to those individuals. In purple, I'm showing you the result for the patients for the healthy volunteers that were taking Reneo, and in the gray bar, you see the placebo individuals. The people who took placebo lost more muscle strength than the ones that received Reneo. And at week 21, at day 21, 1 week later, well, the patients these individuals were already moving and they were regaining their strength. And in fact, the people who were on the placebo regained their strength, but the people who took the Reneo even had a better improvement in strength in their muscles. And what I'm showing you in the right side here is that once we removed the cast, we took muscle biopsies on several days on these individuals. And what we show here is the change in the expression of genes compared to placebo in the volunteers that received Reneo. And you can see that there was nice activation of at least 3 genes. I'm showing them in 3 different colors there. These are genes that are involved in either formation of new mitochondria or in the generation of energy by the mitochondria. So this was pretty exciting to us. And we then went to patients with PMM. And this is the design of the study in patients with PMM. This was an open label study, meaning all the patients received the medication. And we gave Reneo once a day for a total of 12 weeks. And the patients had the option to continue on treatment after that for up to 36 weeks more. Unfortunately, we were hit by COVID, so we were not able to complete the 36 weeks extension, and the maximum duration of treatment that patients had in this trial was about 40 weeks. But 40 weeks is a pretty nice long duration of treatment. In general, the drug was very well tolerated in the patients with PMM. We saw important improvements in the walk tests and in other exercise tests in these patients. We also saw improvements in the symptoms and in the quality of life reported by these patients. And the majority of patients who completed the first 12 weeks elected to continue into the extension study, meaning that they felt that the drug was helping them. Now, the data that we got from this study allowed us to design the study that we are now doing. This is a larger clinical trial. We call it the STRIDE study. And this is a double-blind placebo-controlled trial. And this is in adult patients with PMM who have a myopathy and who have specifically mitochondrial gene defects. The study is going to be performed in multiple centers in the United States, in Europe, in New Zealand, and in Australia. And it's a 6 months trial where patients are randomized to receive either Reneo or placebo. And we monitor, again, the safety first and foremost, but also we look at measures of efficacy, such as the walk tests and symptoms in the patients. At the end of this trial, in the United States, we will not be able to offer an extension study, but in other countries, we may be able to offer an extension study as well. We're currently recruiting from this trial. We're actively recruiting for the trial already in the United States. So we're very excited about this. So what I want to conclude by saying is that I think there is a very strong rationale to test Reneo in patients with primary mitochondrial myopathies, as I show you data. The STRIDE study is open for enrollment in the United States. It will soon also be available in the UK, in Europe, Australia, and New Zealand. And what can I say? We ask for your support, and we ask you to talk to your doctor to see if STRIDE is a good option for you. I want to thank you for your attention, and I want to thank the other panelists, because I think that together we are offering multiple opportunities for patients with PMM to address their diseases.
Thank you, Alex. And I second that. I want to thank the panelists of this session for sharing their updates. It's really exciting to see the amount of activity that's been taking place and soon will be taking place as we move out of this pandemic period. I think I speak for everyone in the mitochondrial disease community. We're really excited about hearing updates on all of these activities in the coming years and months and years, excuse me. So with that, I'm going to ask our panelists to turn off their cameras and exit the stage, so to speak, and then ask Dr. Richard Haas to come on and join me. Richard, we have 5-10 minutes to kind of talk about where things are at in the mitochondrial disease space, with a particular emphasis around getting patients involved in clinical trials, right? So through these panel discussions, we're really hearing about an unprecedented level of clinical research activity that's poised to get underway in mitochondrial disease. You and I have talked about this on numerous occasions. To date, it's never really been a problem getting patients engaged. They're highly motivated to be involved in clinical research. Of course, we need that to continue, but what are your thoughts on engaging patients in the clinical trial process, and how best to make sure they understand the opportunity that's associated with it?
Well, thanks, Phil. No, I think it is really a very exciting time to have so many companies really dedicating their research to mitochondrial disease. Where we are at the moment is that all of these trials that we've been talking about today are really for primary mitochondrial myopathy, which is this PMM abbreviation.
Not all of them, but several of them for sure.
Well, no, the ones that we're the group that we're talking about today do not seem to affect the brain, they affect the muscle.
Correct.
And certainly in the future, we're very much hoping that we're going to have other treatment treatments available that are going to work for the brain, for instance. But in terms of patient recruitment, these particular trials really require that there are muscle symptoms involved. And they are really looking at the ability of the patients to walk, or at least to use their muscles. Some of the disorders are so severe, like the TK2 patients, that the majority of them are not able to walk. But we're still looking at muscle effects of these treatments. So in terms of what's available to patients, I think the first thing is that they need to talk to their own doctors, hopefully mitochondrial specialists who are involved with their care, who can just tell them what the lay of the land is at any particular time. And then if they qualify for a treatment trial, one of the most important things is not just their symptoms, but whether they have a molecularly proven diagnosis.
Yes, yes.
And unfortunately, as you well know, we have a lot of patients with mitochondrial disease, and we don't yet know what their genetic defect is. So it's very helpful with some of the trials. Stealth has done this in the past, and I think Astellas is prepared to do it. And also TK2 to offer genetic screening. So that's an opportunity for patients who have, in this case, muscle symptoms, but don't know their genetic defect.
Yeah, no, you've mentioned a couple of really key points there. You know, one, there's still a huge unmet medical need around brain penetrant drugs and how we treat the pediatric population that has so many neurological symptoms in mitochondrial disease. We know there's things being worked on. You know, of course, that's a bigger challenge to be able to deliver therapeutics to the central nervous system. But we're really hopeful that more will be coming on that front soon. But for these patients that have primarily muscle-based symptoms, for the first time, they may be looking at several options, right? And there may be a tendency to want to shop that opportunity. Which clinical trials, should I be involved with? And as you mentioned, no patient should feel like they're on their own in making that decision. Yes, there are online resources like the UMDF website, clinicaltrials.gov, but their healthcare team is a really important support team, right, to have a discussion about which trial is best for them, because it is going to be a function of very specific inclusion, exclusion criteria. And really only the doctors that are taking care of them can help make that decision. Isn't that right?
Yeah, that's really important. And location is important too.
Of course.
We don't really want patients flying from New York to San Diego. They need to find hopefully a treatment site closer to them.
Yeah, and that's not just for convenience, it's because it could fundamentally change their health, right, as they prepare to enter the trial.
That's correct. It's very stressful to have to travel a long way. And so with a number of the trials, we're emphasizing that the patients need to have a day of rest, basically, before they start in on the testing. Yeah. And of course, they have to have a caregiver accompanying them. And there are lots of nuts and bolts associated with the with having an organization that can help with that whole business of actually getting to the site and then where you're going to be while you are at the site,
right?
All arrangements, travel arrangements, and so on. And just to run through the way that these things work, often there's a pre-screening opportunity where the physicians caring for the patient are kind of involved with them in deciding, as you say, whether they might be suitable for a particular trial. And in some cases, we've had a registry with the stealth studies that went by the number of 300. So that was their trial 300. And that actually helped them identify patients who would be suitable for the trial. So that was a kind of run-in pre-screening. And that's followed if that seems an appropriate trial for our patients, for them to undergo a screening visit on site, which is a much more complex, often whole day event, when they're going to be evaluated in detail and have blood and often urine testing done, and review of their medical record and their genetic diagnosis, the genetic findings. And then the investigators at the site have to tick off whether they meet all of the inclusion criteria and they don't have any of the exclusionary criteria. And that's something that we pay a lot of attention to now, even in the pre-screening phase, because we don't want to bring patients from a considerable distance away and then see that they're just going to fail entry criteria. So there's a lot of to and fro going on before you actually start on the clinical trial, and that's where our clinical trial coordinators are essential.
Yeah, that it really does take a team, right, to execute on these clinical trials. And I think if there's a take-home message from all of this, options are a good thing. Now that multiple companies are moving forward, multiple potential interventions, that's more research questions being asked, because that's really what a clinical trial is about. It's a clinically oriented research question. Is this drug effective? Is it safe? To the extent that those happen in parallel and that we get answers more quickly, then we have a better chance of getting to meaningful therapeutics for our patients. So, I really hope everybody leaves this session with a determination to consider whether they could be a participant in a clinical trial, because that is what it will take for us to have treatments and cures for mitochondrial disease.
Yeah, and I want to emphasize that in terms of knowing what's going on, the mitochondrial specialist, if they have access to one, can certainly help. But the patient-focused organizations like the UMDF are critically important in providing information. And then the clinicaltrials.gov website is a bit more complicated, because it's kind of like the internet. Things that have finished or closed are still on there, and you certainly can search for mitochondrial disease, but it's a little bit hard to interpret on your own. And actually, as somebody who has had to deal with entering data into the site, it's particularly difficult for us as well.
Sure. Yeah, no, really good reminders for our community. We're going to need to wrap it up. We're at time. Dr. Haas, can't thank you enough for how you've taken care of our mitochondrial disease patients for so many years, and also your deep involvement in these clinical trials and really trying to bring these treatments and cures to market in an effective way. So thank you for that. Thank you for your comments today. And once more, thank you to all the panelists that were involved too. After a brief break, we will be back with a discussion panel focused on genetic testing. And I think we have some really exciting insights to share with you as well. Thanks, everybody.
Thank you, Phil. Thanks.